speak about biodynamics, fundamental approaches and applications, which I think is very important to connect these two things.
Well, thank you very much for being here. So it's a sort of resilience, because unfortunately we could not organize any graphically, even though I think that all of us we it's a pleasure for me to talk about what we are doing, especially being surrounded by friends. So I will shortly present the core principle, the motivation behind our study, the methods that we are lately developing, some applications. I will pinpoint the most important results and, of course, a summary. Well, you could easily recognize me. And the reason for doing this is that my face is large enough. Unfortunately, bacteria are pretty tiny, and not only their size, but also the Abbe limit make microbial identification just from a single label-free imaging pretty challenging. So what should we do? So the idea is that why use just an image and not a movie to try to gain some information and eventually specificity? So the idea is that we have to develop a sort of multi-dimensional time-lapse assays with pretty high resolution to extract some quantitative measures. But what I would like to emphasize is the fact that we are interested not just to relate the specimen to the time-lapse of the parameters that are directly measured, observables, but to try to solve the inverse problem and relate, to characterize the system that we are interested in by analyzing the dynamics of its parameters. It's pretty challenging, and I'll try to present this as clear as possible. So the idea is to derive some biophysical parameters out of the movie, and we are doing this not by performing optical measurements, but electrically assisted optical microscopy. And well, just for you to have an idea, you will have some, let's say, time series that we are trying to analyze. For me, I was a little bit upset by the fact that the dynamics itself is not such spectacular, you know. But the other point was, do we need very fancy equipment to gain the information that we need? And then I remember about something you may most likely know, that, well, of course, initially you need information. Picasso first painted the real bulls, the bulls which with an entire set of details. And lately, he managed to get to the let's say, more simplified to reach the essence without simplifying. And with this idea in mind, the problem is that, well, why if instead of just thinking about image, to try to get the dynamic information out of this, just to try to make things a bit more clear. The problem is that we would like to use, of course, pretty sophisticated methods in order to gain as much information as possible, and then to analyze which one is really the one that is essential. So that was for me, that was the idea behind this comparison, of course, with Picasso. I'm far from his skills. Well, of course, we are motivated by by trying to address real-life problems, biomedical, food, environmental, and so forth, and to translate them into scientific problems also, of course, trying to find some practical solutions. Well, tomorrow we'll have some time to present the results that were developed in our center. Christina was mastering part of care detection system for biomarkers. Sorin will present the multi-channel system for capturing, assess the viability, and also perform the rapid phenotypic antimicrobial susceptibility or resistance testing. But for now, what I wanted to pinpoint is the fact that we are interested in high resolution single cell time-lapse assays, and more than 3 decades I was very fond of electrical impedance spectroscopy. Unfortunately, this is a very used technique for biomedical application, but it lacks spatial resolution. You cannot properly see a single cell, although the information electrical impedance spectroscopy is able to provide quantitative information related to the integrity of membrane. So the idea is that this is a way to gain information about cell viability. The point was that our group was pretty interested in to derive electrical impedance maps with optical resolution. So that's actually the message that I wanted as a take-home one. Well, a couple of years ago, together with Gabriel Popescu, we developed a system able to provide high-resolution impedance maps. And what I wanted you to see is the complementarity of the optical images versus the electrical one. So in the optical message, there's practically no difference. You cannot assess any difference between conductive or not conductive areas, whereas it's a clear information provided via the electrical images. Well, we played a little bit with the frequency and performed in parallel the same type of experiment using the standard electrochemical impedance instrumentation, and this one and the parameters that we derived were pretty much the same. Well, so the idea is, okay, so besides optical information, we are able to monitor multidimensional, meaning especially electrical and optical parameters in order to provide single cell analysis. But I was very much interested in solving the inverse problem. For me, that was the challenge of the last couple of years. Tomorrow, Mihaela will present the experimental results, the relevance of this endeavor, and what type of information, of cellular information, is able to be derived using these methods, also related to the closure of the gap junctions. What I just wanted to tell you is that this information is complementary to the standard optical one, and this is label free. So that's let's say the good news, and I don't know bad news for this. Well, just a little bit several equations just for you to have the idea. Well, what's the point? Which is the core of this information? optics means interference between a reference and an object field. That's something everybody knows. What we are doing still is the fact that we are, so this is what got it published a couple of years ago. The point is that what we are doing is to apply an alternative current, so to play with this term, and essentially what we measure is the intensity, which is related to the frequency of the alternative field and the electrical parameters of the sample. So that's the whole theory that I was that I wanted to pinpoint. But related to this, there are two different things, and the physics is slightly different. So whenever the cell, the specimen, is adhered onto a cover slip, then what we measure actually relates to its shadow onto the electrode. So from the electrical perspective, what we measure is the modulation, the perturbation of the complex dielectric constant of the Thomas Fermi layer within the transparent electrode. Is not the cell itself. So this is, so everything is encrypted here in this dielectric constant and its fluctuation. But when the specimen is above the cover slip, what we actually measure is the cell. It is the cell which provides the information that we want. And we can do it by entrapping the cell in a hydrogel or by using magnetic particles. And what I think is important, and this is the real take-home message, is the fact that because the effect of the electric field relates to the optical parameters that are measured, any optical method can provide the same information, well, more or less. But any optical method, including optical coherence tomography. So actually, Christina will provide the information on using electrically modulated optical coherence tomography tomorrow. Just to pinpoint the difference between the electrical and optical properties, so you can see here by the contrast in this is an optical that the bacteria has a larger contrast and the magnetic beads are almost transparent, whereas it's exactly opposite because their conductivity is far lower, the bacteria versus the magnetic particles. So you can easily, from this perspective at least, separate them. Well, of course, what optics provides is a capability to measure population, dynamics of population at single cell level. And this is what we measured in case of cells, magnetically labeled cells. And this is the information provided by analyzing the evolution of the population in these cases. Well, the problem is that whenever we talk about population, we gain, of course, average information. And what I would like to point out is that these techniques provide information at single cell, and I will emphasize this. So the most important thing, and that was actually the information related to the title, is the capability to assess the viability of bacteria. And this is accomplished just due to the fact that viability loss relates to decrease of the membrane impedance, and this relates to a decrease of the electrical contrast. And let's show you a result that will hopefully convince you that this is the case. So what we did was to play with choline. Eric also presented some results on biofilm. But in this case, what you see is that after 48 minutes, well, there is a difference, but not too significant in between the contrast, the optical image or the optical images due to the exposure to antibiotics. Whenever, for the electrical images, in about 12 minutes, you already see the difference. So it's, for me, was wow. So this means that without any labeling, it is possible to gain quantitative information about the viability of a cell, and you can do this clearly at single cell level. Well, after that, I said, okay, let's look at the entire population. And I marked here the cells that I presented in the previous slide. What we see is that, well, we are not very happy, but at least for apparently low concentration of antibiotics like choline, there is a heterogeneous response of cells to antibiotics. Clearly, not all bacteria respond the same. So there are plenty of others which don't care. In our discussion yesterday said, well, perhaps it has to be something related to the fact that the bacteria were in various cycles of the cell cycle. Unfortunately, we cannot control, we cannot synchronize cells, bacteria, if we are infected. So we have to think of various others way to cope with this heterogeneity. The idea is that in some case, we may rely on the actual observation, so you may not necessarily need to solve the inverse problem in order to gain some information as the one related to antimicrobial susceptibility testing. But it could be of interest to try to do this. And again, that was and the problem of my last 2 years. And just for you to have an idea, what is it about? Gradient light interference microscopy, a method developed by Gabriel Popescu, provides pretty fast, accurate quantitative phase. And actually, it is based on phase shifting, which of course takes time. And well, it's not entirely cheap. What we did was to perform these type of experiments, of course, with electrical actuation. The phase, in this case, the phase difference is simply derived based on the phase shifts. But we were interested to find out if just merely by AC, alternative current actuation, we could get rid of the phase shift. And in my the last slides, I'll pinpoint the results. So what we did was to measure using this method a cell which was fixed on top of an indium tin oxide electrode, and that's the result used provided by optical measurements and by performing the Fourier analysis of the time series of the intensities measured on each pixel. We got these pretty nasty images, but that was by that time. And well, then we managed to derive also the electrical phase shift maps, which look a little bit more interesting, let's say. From these, we solved the inverse problem and derived these maps. They are not looking nice, but this is the first map that provides information that these can be solvable. It's so you clearly cannot be impressed by the quality of this image, but for us was important because we could show the proof of concept. And what is the most important thing is that this is how the image, unfortunately, doesn't look nice on the screen, provided by the standard gleam method on the left, and on the right is the image provided by via solving the inverse problem. And for us, of course, there is no cosmetics here. So these are just the data that we obtained. So for me, this was important because the challenge was to have a clear information that it is possible to obtain the same type of information not by phase shifting, but via electrical actuation. So it is not the fact that it is simpler, it perhaps even cheaper, but it provides more information because it allows you to solve the inverse problem. So it is not entirely trivial. Well, with this, I would like to summarize. So the idea is that AC electrical actuation enables quantitative optical microscopy, similar with the time shifting for QPM, to recover intrinsic sample parameters like the optical or electrical parameters, and well, while simplifying the experimental setup. Electrical impedance maps, which I think that's actually the most important result that I wanted to show you, provides quantitative intracellular contrast revealing distinct cellular features and the viability. So the fact that it is possible in a couple of minutes to gain information about the alteration of cellular viability without labels, well, this was a result that for at least made me thinking that that could be a way to go. And it is important, again, not only the fact that this was possible to be seen by analyzing bacteria, but by analyzing organoids or spheroids using OCT, the same type of information is available. So that's why I think it could be a way to go. Well, last but not the least, electrical actuated optical microscopy supports quantitative phenotypic drug susceptibility assays in microbial and tumor cell population, resolving heterogeneous responses to at single cell level. With this, I would like to acknowledge the support from the Ministry of Research, Gabi Popescu, a very good friend of us who unfortunately passed away, my colleagues from the International Center of Biodynamics who worked pretty hard, and of course my old friend Eugen Gheorghiu Romeo from Paris. And with this, I would like to thank you for your attention, having in mind that the reason why we are here is to develop any type of bridges. You may remember that such series of bridges are in the summer in Beijing in the Summer Palace. So that's a picture that I took some years ago. And well, the world is not very large to not to allow us to shake hands. Yes. Thank you so much.
Please, questions if you have.
Very detailed question. The last image is a show where you decided to map the directed constant. What does it actually mean? Because it's not one directed constant, it's a pixel there, right? It's a cellular image.
Yeah, well, of course. So actually, this relates more to the optical path, of course.
Okay, so it's not
Any other question, please?
maybe in the beginning you thought like wanting to create a lot of information, so dynamic information. So in the end, you want to do that.
Well, the idea was that, that was about the principle, and then the interest was, can we really go that pathway? That means, because the point is not just to rely on what we actually measure, but on the intrinsic properties of the cells. So the big question is, can we derive, can we solve the interest problem to get information about the intrinsic parameters of the cell? And this is pretty challenging. So that was actually the reason that I put this by the end of the presentation to pinpoint the fact that, well, it's not easy, but it's doable. Yes. So next presentation, I would like to show the movie of these intrinsic parameters and related to Picasso, just to show you a movie taken using, well, let's say, pretty excellent objectives able to correct any aberration and simple ones, because I do believe that there is no need when following evolution to use very precise information. The dynamics should have the same features. So actually that's
So practically, we would like to, what you said in the time of the talk, we would like to have like a rapid assay that can be used to estimate the material resistance, which ones are in what stage. So that's the goal, right?
and the tool should be simple.
Yeah, so that then it can be applied in any medical kind of institution for diagnostic activities.
With a fancy QPI microscopy to be
Right, I think this is very important. Anybody else has a question or a comment, please?
well, we are just in the first day of it was quite a long day.
So it was a whole day, but I mean, it was very informative for me and somehow diverse, but you can intellectually put it all together and relate. And I think that was for me very exciting. Somehow there was always good to see the links and an exchange of the knowledge which each of one can use and to implement in our own niche. So thank you very much for everybody.